Thank you so much for being here. Well, welcome to this first webinar of the Working Group on Ecosystem Services. I would like to thank the Secretariat of GEO BON for helping on the organization of this webinar. And why a webinar on eDNA and molecular DNA? Because, well, for obvious reasons, they are it's an emergent technology over the last years with lots of opportunities, promising results, but also that puzzles lots of people too, and puts many people to ask about limitations, and some of them are also very afraid of the different products that can come out from these different technologies. And especially in the scope of ecosystem services, many of us had already discussed the potentialities of eDNA and related methods and technologies, but especially for us more conventional ecologists and more conventional social ecologists and social scientists and practitioners, we lack this clarity or a materiality of the things that eDNA can offer to unravel the different dimensions of biodiversity and the opportunities in time and in space that it provides in a complementary way to other strategies and to other methods. So with this, I would like to also give a special thanks to our speakers, Karen, Zacchaeus, Paul, and João. They will be giving us their slight bit of their experience in a 5-minute conversation, a 5-minute presentation to help us establishing here some food for thought. So with this, I will introduce Karen Dyson from the Dendrolytics in Seattle, United States of America, and also Spatial Informatics Group from Pleasanton, California. And she will be giving us an overview of her experience with soil sample-based eDNA as a monitoring approach. So, Karen, the stage is yours.
Thank you so much. Let me just share my screen here. There we go. Everything good?
Everything perfect.
Yes. Hello, everyone. It's so great to be here. I'm really excited, and I wish we had more time to geek out about eDNA. My name is Karen Dyson, and I run the eDNA portion of a monitoring tool called TerraBio, and we use soil sampling in order to help monitor private investment in sustainable agriculture and development. So these are things like silviculture, forest regeneration, non-timber forest products. And we have really had a lot of success using eDNA, and we found that it complements our wall-to-wall remote sensing by quantifying biodiversity in specific locations. For this tool, TerraBio, we did our pilot in 2021, and we now have 2 projects onboarded in 2022, and we're looking forward to a very busy 2023. And we've successfully evaluated diversity on farms with agricultural investments and compared these to what we call business as usual, which is usually a pasture, as well as forested and regenerating alternatives. So I'd like to frame my comments today in terms of trade-offs. We've found that there are often multiple ways to do something that might be, I'll say, scientifically acceptable, but more or less useful for your particular use case. So for us, we need to collect soil in a way that's repeatable and robust, easy for the field technicians, and provides good data. And easy for the field technicians, I'm just going to highlight real quick, because of how we work with private sector, frequently the people who are helping us collect the data are actually the farmers themselves. So they are local people without they haven't gone to school for, how to do field work. So we need to have something that's approachable. We actually started with a standard plot design. This is a very common ecological design used in lots of peer-reviewed studies. But we also found it was very expensive to set up and hard to do. It was very hard to find the precise locations in the field where you're remote without necessarily very good GIS positioning. So what we ended up doing was moving to it's like a homogenized bag where it's this, I hope you can see my cursor. It's the second image here. Where the data collectors can actually basically scoop many, many, many different pieces of soil from all over the field. And it is all collected in one bag, and it's about a couple liters of soil. And so we then homogenize that bag and then take samples from there. So the replicates there become what we analyze. And so this actually lets us keep the farm as both the unit of observation and the unit of analysis. And we've also found that it reduces the personnel cost and is much simpler for our people on the ground. And as a side note, we've also found that collecting these higher soil volumes has actually given us a lot better data on the back end. So we're really excited about kind of trying these non-traditional approaches. I also want to share another type of trade-off, which is for the, how we present our results. And TerraBio uses indicators to communicate our results to stakeholders. So this includes investors, as well as the farmers themselves, and helps give them a metric of how well they're doing, as well as kind of holding the investments accountable. So we need indicators that are robust and repeatable, that are easy for non-experts to understand, and that are scientifically accurate. So eDNA has some unique data properties, and we need to be able to present indicators that respect those data properties. And we also need to be mindful of the fact that eDNA is actually kind of two pieces. So you have the data that you collect from the field, but if you have to do any sort of taxonomic matching, you're also you're querying a database that matches the DNA snippet that you get with a taxonomic identifier. So if you need a lot of taxonomic information for your indicators, you're actually relying on these two different pieces. So this is definitely a challenge, but one that we've really enjoyed tackling and have been able to find some solutions, and we're hoping to improve them in the future. So one of the things that we found is that non-experts frequently want familiar indicators, so things like key species detection, but eDNA really excels at rapid community metrics, so we've had to balance those. We found that diversity metrics have been really successful for measuring biodiversity and distance between communities, especially so. We're hoping to, in the future, improve some of our ecosystem services beyond biodiversity, for example, pollination. But that, again, is reliant on some of those explicit taxonomic identifiers. So that's a little bit work in progress. But even with just looking at like arthropods broadly, you still need that one piece. So you need to have at least enough identification to identify that broad group. So that's definitely an ongoing challenge. So I will end it there because I think I'm out of time and pass it back to you.
Well, thank you so much, Karen. Yes, as far as I could understand, there is lots of clients or end users, especially in the private sector. Yeah. But then the great challenge is how to give a reply to all these analyses, especially when it comes to make a match between the taxonomy and the DNA that is collected through the soils. This is lovely, Karen. I think we're going to leave the questions for for the end of the different presentations. Our next speaker is Zacchaeus Compson. Is coming from the Advanced Environmental Research Institute from the University of North Texas in Denton, USA. And Zacchaeus is going to share his experience with eDNA as a bioassessment tool for local stakeholders. Thank you, Zacchaeus. Go ahead.
Thank you for the introduction. So yeah, I recently started as an assistant professor at the University of North Texas, and have started my own environmental genomics lab where we specialize in DNA metabarcoding. But importantly, I did not start out using eDNA. I was trained as an ecologist, and as such, I've kind of like had to wade my way through all of this new information to use this new tool. And so if you're to take away anything today in the next 5 minutes from my talk, it would be one, you can use eDNA and perhaps you actually do some of the techniques. And secondly, and perhaps more importantly to my part of this talk, you can utilize a citizen scientist to greatly enhance your sampling and science capacity. And so in a recent review that we did on metabarcoding, which is the use case of DNA, we surveyed all the literature we could find using this approach and found that, of course, it's a growing technology that's occurring all over the world and occurring, as you can see, across all these different taxonomic groups around the world. What's interesting is when you break down the data from this review, you find that that in general, the government and academic sectors have dominated this research, and with much lesser amounts coming from commercial, NGO, and industry, although there is an uptick in some of this in recent years as you're getting this explosion of information. But what's not seen in this graph, and what's hard to capture from these papers, is how much this rise in capacity that you see in recent years, or rise in use of these techniques, is actually enabled because of citizen scientists. And so citizen science is a powerful tool for many reasons, and we don't have time to go into them all, but one of the biggest reasons is that it rapidly enhances your ability to collect huge amounts of data at large scales. And by mobilizing citizens, it also enables these rapid responses to things like species invasions or detection of species of concern that might be rare or endangered or in threat. But the other kind of intangible here that is a huge benefit is that by engaging citizen scientists, we actually can help to draw connections with citizens and their local environments, and then give them kind of ownership to be stewards of what's in their backyard. And we can do this through formal programs, such as some of the ones you see on the left that are reviewed in this paper by Larson et al. But you can create these basically, citizen science sampling packs or sampling kits, and then with a little bit of training, they can actually collect a lot of information. So what I'm going to do now, instead of just talk on a high level about citizen science, is go into 3 specific case studies that I think more than talk about, that citizen science is good, this shows that they actually can be used. And so when I started out as a grad student engaging in citizen science, it was primarily just to give citizens in the community exposure and school children exposure to science. I never was thinking about the potential of them to be true partners that could actually collect vast amounts of information. That was until my first postdoc, where I and several other postdocs network with other 9 school teachers across the southwestern United States and collect, and using them, these teachers, many of which you can see on this paper as co-authors, were able to collect 500 samples across 100 sites. Now, that's a tremendous amount of work that I could never have done. In fact, I collected 0 samples for this project, despite doing a lot of work on it and coordinating with these teachers who were very motivated, took their job seriously, and really got to show their students some awesome, like, hands-on science. And from this, we were able to do really great science. For example, in this paper, we're able to show that that despite us having like these very different deserts in the Southwest, there's a lot of shared taxa, and then some key unique taxa that vary across these deserts. The other thing that we found that was really interesting is that climate primarily works through these secondary variables in an indirect way to influence microbial communities. The point here is to not get into the details, but just point out that this is a group mainly of citizen scientists that not only collected data, but worked with us through the whole process, all the way through publication into a peer-reviewed journal article. So the second study is a project that I got involved with when I was working with Donald Baird and Mehrdad Hajibabai, who are some of the fundamental, or some of the first people that started advancing DNA metabarcoding as a tool. And this project was born out of necessity, in a sense. So there was a report that the WWF of Canada put out that showed basically very poor coverage of aquatic knowledge about invertebrates across Canada, which is a very large geographic area. And even worse, huge data gaps in most of Canada. In fact, this got such attention that it was brought up by the Prime Minister Justin Trudeau and made it into a lot of local watersheds, because the watersheds are in trouble, but we also don't know a lot about them. And so this basically led to what we call STREAM, which is sequencing the rivers for environmental assessment and monitoring. And basically what this is a partnership between Environment and Climate Change Canada, the University of Guelph, and WWF, and then a huge amount of other stakeholders ranging from Indigenous groups to school groups to other government groups across Canada. Now, this, of course, doesn't cover all of Canada. A lot of Canada, there aren't many people. It's very not very densely populated as you move away. But this coverage has enabled us to collect, just in the first round of sampling, and by us, I'm talking about citizen scientists who were formally trained and given these sampling packs to collect over this huge geographic range. And once again, do really cool things with the information, including methods testing. As you can see here, testing whether the water, the DNA we get from the water and the DNA we get from the benthos give us the same diversity signal. And you know, you can look at the paper for more details, but the point is, this is one of many papers coming out of this project. We also were able to, in this project, test many different primers. So getting the samples often is very hard and costly. Once we have those samples, we can do so many cool things. And some of these questions that this group is answering are at the cutting edge of DNA methods and what we know about metabarcoding. These questions about how many primers do you need to recover your diversity are fundamental and have sprung out of the fact that we need big amounts of samples to actually answer these. And the last case study takes all of this close to home and just shows how on a slightly more small scale, a local scale, you can really generate awareness about biodiversity in your backyard, as well as good information about that biodiversity. So here we have one of my graduate students, Haley Daniels, who as an undergrad, loved citizen science. She was involved with iNaturalist. She was involved with all of these citizen science things. And she really has championed trying to use citizens as scientists that are collaborators on her project. On the left, you see the olive shaded bird dropping moth, as it's so kindly referred to, or as I like to call it, the disco moth. But this is one of many species that were found in these bioblitzes that Haley started hosting well before she was in my lab. And so what's amazing about this study to me is, if you look on the left, you can see that this environmental area is a relatively small patch of green space in a huge DFW metroplex. And as you zoom in, it's quite a large area. It's like around 2,500 acres, but in the context of the sea of urbanization around it, it's very small. Now, just from her citizen scientist work, Haley was able to basically collect across not just moths, but all these different groups, information that brought this public record through iNaturalist from less than around 100 observations and very few taxa to over nearly 3,500 records of 777 species. And as you can see, the green numbers here are the amount of citizen scientists that were at these bioblitzing events. And in just a few events with a lot of people and a few experts around, they were able to get this record vastly enhanced. A, it's just amazing there's that many species in an urbanizing area. And B, you just don't know what's there until you actually look. Now, the cool thing is, as we're working with all these citizen scientists, we're thinking about all the other research Haley's doing. And one of the ideas we had that the citizen scientists are super stoked about is, what if we could collect the DNA that's collected on these sheets when we do this night trapping? And so that's what we're working to do now, which will then help to connect the observations that the public are making in huge numbers with huge amount of participation to fundamental records of DNA that we can then use to create barcodes to enhance our local libraries for future bioassessment efforts. And so I'm just going to close with this slide that just kind of shows a new paradigm that was put forth by Larson et al. In a Frontiers in Ecology and Environment paper that recently came out. And basically what this shows is you've got your citizen scientists that are networking on the first arrow on one that are enhancing the ability of a scientist depicted using the computer to actually gather biodiversity data, and then coupling that with new techniques like Earth observation approaches, as well as on-the-ground estimates of eDNA, also enabled through citizen science collection. We can actually vastly expand our reach, which is essential for us to do in this time where human impacts in the Anthropocene are rapidly changing the world. So this effort is not only possible, but I think it's essential. So with that, I'll take any questions later, but I want to thank all the awesome citizen scientists that helped with these many projects. Thank you.
Thank you so much, Zacchaeus. You gave a very nice overview on also different applications of collecting eDNA from water, soil, and from this more insect view into the urban system as well. And this is an additional challenge, right, to involve people as well and citizens. Karen already showed the involvement of local stakeholders, and with you to bring common citizens to embrace these novel technologies. So now we will move to Paul Henning Krogh from the Faculty of Technical Sciences in the Aarhus University in Denmark, who is going to make a brief, try to get into the 5-minute timing about soil eDNA for biodiversity assessment and soil ecosystem services. Thank you, Paul.
Thank you, João. I'll just start sharing my screen, which we tested once, but let's see if I can make it work. Thank you for inviting me and giving me the opportunity. Just a second. I'm afraid I have some problem now for some reason.
Yeah, don't worry, Paul. If you need some time to get the problem solved, maybe I can challenge João to go next, if you agree, Paul.
That's all right.
Yes. So we also have João Silva Rodrigues from SGS, Global Biosciences Center in Lisbon, Portugal. And João is going to give us an overview on the application of eDNA monitoring in support of the business sector. So João, hopefully you will be able to share your screen. And
yeah.
The stage is yours. Yes. Perfect.
Can you see the screen? Yeah.
Yes.
Yeah, that's fine. Yeah. So thank you very much. Thank you for the invitation. A really good opportunity to really show that I think eDNA is already, and we believe as a company, on the private sector, that is a robust technique that can be applied worldwide. And we're seeing, as Zach was mentioning, the uptake from academia now to the business sector. And what I really want to show you today is why we believe in this, why we're putting all our bets as well on this, and why we believe it's essential to do it. I'll bring you some use cases today. So for us, we are really, as Jesse, the largest testing and inspection and certification company in the world, and I think it makes sense that we also enter this space and we provide our clients with all this. So we are global. We have more than 2,000 labs across the world, but I won't bother with our corporate jargon today. More to show you that we want to put eDNA across the world everywhere, so that it's accessible to everyone, to citizens, like Zach was showing, to academia, to businesses all over the place. We really believe, because we are a purpose-driven company, we wanted to make and create a better, safer, and more interconnected world, and this is part of it. We are, in a way, and that's why we've created this Center of Excellence, and we've recruited a lot of PhD-trained scientists to work here, because we face a lot of ecological challenges ahead, and eDNA, microbiome analysis of soils and all that is part of the solution for sure. I don't think I need to explain to you all that we are in deep trouble, and we believe as well corporate business know it well, and besides governments putting targets or whatever, I think it's at the end businesses have a role to play. And we see that we're getting requests of proposals almost every day from business worldwide to know more, to start tracking what they do, what they are doing harm to in their operations in a way. And this is part of their sustainability strategies, but also showing that actually we're not doing harm to the planet and we can contribute to a better planet. We've started looking at eDNA because as a testing company, we believe DNA-based approaches are the best. After reviewing most of the techniques available, we want to integrate it with others like satellite, like many other tools. But really, I think if we want to do this at scale, I think we all agree that we need it. We cannot rely just on taxonomic expertise on site everywhere in the world, and we need something that is standard and reproducible. So really, we saw it's really non-invasive, and we have increased resolution and higher accuracy as well. So this is crucial. We're doing it all sorts of, as you've seen, water, soil, air, and many other. So we've tested and optimized all this, and we can offer all this to our clients globally now, together with targeted, etc. And comprehensive reports, that's essential. What I wanted to show you is really some use cases to show that we are applying this. As I said, we're doing it regularly now across the world. In the aquatic environment, we used it, for example, recently on a huge marine protected area in Cape Verde, where we supported conventional surveys with divers with eDNA as well. And we really believe that it's so powerful, it's complementary, and above all, less risk, and enable us to do it in a larger scale. And just to give you an example, in this area, is now being proposed to be a new marine protected area, the eDNA results on its own pick up more than double the fish species that were recorded during all the dives, all the cameras. And really, when we go into benthic organisms, invertebrates, and elusive species, it's a complete different world that is not picked up by any of the conventional techniques. So really complementary techniques that will enable us to do this at scale. On the other side, bringing something that's probably already been shown on the urban side, and it's probably one of the biggest opportunities we have in the built environments to bring biodiversity back in scale as well, is really insect populations and how cities can play a role. And we've been looking at that quite closely, and we've come up with this insect scan that we're now, and it's really the thing that we're doing that has the biggest uptake, which is really to look at gardens, at vegetation, and start seeing and mapping all the species that are present that have visited these sites. And we're now offering this worldwide, and then really taking off quite well. Just to show you, this is our office outside. We just picked some vegetation, some flowers, and in 15 flower samples, we picked up more than 68 species. And also the manure that was passing by, we can discuss that. So from pests, pollinators, all sorts of, And so we've done that in cities as well, optimizing mowing strategies just by getting vegetation. And from bags of 1 liter of clippings, we can go up to 100 species of insects. And you can see here in this chronoplot some of the results and of the relative percentage of this species, and how we can then start informing across different parks, like we're doing here in the city of Nijmegen in the Netherlands, how their strategies to mow, their strategies in urban gardens, all across. So we can really start having a look and compare practices a long time. Also, and as you can see here, even functions and pollinators and pests can be mapped, and all the ecological metrics can be then used. And this, we believe, it's the basis of starting to be able to do and implement nature-based solutions and bringing ecosystem service to practice with the real data gathered along the time. And just to finish up, a joint project with a startup in Portugal, NBI, which we looked at almond trees and pollinators, and just to start seeing if we could, by looking at flowers, to see in different fields with different types of treatments and different ecological practices in a way, if you see some of the differences. And you can see here some results between the different fields, and also if you look at individual flowers, you can almost start getting a feeling of how many are visited actually by pollinators. And you can see here in this, for example, that only 21% of the flowers, and that's something we can discuss in terms of quantitation and the power that eDNA has for that or not, but that were visited by the honeybee, the Apis mellifera. So this is something that we're starting to do, and really, I think eDNA is going to revolutionize all the way that we can do this in a reproducible way worldwide. So yeah, so just warm up, really looking forward for the discussion. Yeah.
Well, thank you so much, João. It was a very nice overview on how scientific knowledge and research can be together with the business sector to deliver exactly what GEO BON pretends to, which is monitor at a wide scale biodiversity, ecosystem functioning, and ecosystem services. And it was also lovely to see the scale that eDNA can actually bring. So really looking forward for the questions on this. I see Paul already is here keen to present his part. So, but Paul, let me just give the introduction again. Paul Henning Krogh from the Faculty of Technical Sciences at Aarhus University in Denmark. So go ahead, Paul. Thank you so much.
Yes, yeah, thank you for this opportunity. So I just jump into our challenges, which I know some of you are very well informed about. So we need to, I'm talking only about soil biodiversity, right? And I'm talking about eDNA, which when we talk about soil, it's actually DNA in soil is not the organisms as such. That would be bulk DNA or community DNA. But as mentioned before, we need to have the links between all the reads that we get and the species, which means we have to have good reference databases. They are, for some soil organisms, they are not very well developed yet. We need to have the links between DNA read abundances and the biomass, which is, as most of you will, many of you know, this is not completely settled yet, with particularly not for metabarcoding. We need to have the links between the species and the functional traits. And this is very crucial because the functional traits will enable us to calculate ecosystem services. This is a key paradigm for soil biodiversity and ecosystem services. And when we can assess the ecosystem functions from the functional traits, we will be able to finally end up having ecosystem services as the end measurement, you could say. We also have some methodological challenges. I mean, which kind of eDNA extraction should you use? How much soil? This is really a tricky point because we want to sample as little as possible. We don't know much about that. It hasn't been tested out, and this is needed if this way of working should be more common and possibly also standardized. So the whole spatial distribution of samples and the amount of soil you need to take is very crucial, need to be specified. There's very little research on that. Then there's another issue. This is what kind of eDNA do you actually have? Is it old or is it new? This is also very relevant for what you are asking for, what kind of biodiversity you want to know something about. Concerning QA, quality control, we need to settle what kind of mock communities or positive controls for our whole procedure, how to deal with that. So our conceptual framework of workflow is we have some problem we want to solve.
Paul, I'm so sorry for interrupting you, but we are getting here many people asking for a larger screen. I'm not sure if you're able. Yes, we see.
Okay, I thought I had the last screen.
Yeah, larger one. Yeah.
That's then I have to stop share and look again.
So sorry.
I'm sorry too. I thought I had chosen the. So
if that is not possible, we will continue like that.
I wonder what's happening. Why I couldn't have the right PowerPoint. So it simply looks too small.
Yes.
Maybe
but if it is not possible, I'm sure people can.
Sorry, yeah.
So Paul, when you go to share screen, if you pick screen, and then if you on your presentation go to presentation view, then that should be the full screen that we see. That's the easiest way that I could figure out how to do it.
All right. Maybe I'm choosing the wrong screen because I have the two screens actually.
It looks like your screen is vertical. Do you have a vertical monitor?
Exactly, yeah, it's a vertical monitor.
Yeah. That's probably why.
That's the problem.
But that's fine, Paul. Otherwise,
I'm sorry. I don't think I can solve that now. So
let's give it a last try.
Right.
Share the window instead.
Yeah, I could do that.
Stuff public, Paul.
Yes. Now I have to get back into. I'll share the window instead. I think this is bigger, isn't it?
That's nice. Yes.
Excellent.
Let's continue. Excellent.
Although it's. So I'll just move on to. I only have 1 or 2 slides left anyway. So. Yeah. So our conceptual framework when we talk about soil biodiversity is to rely on soil biodiversity databases, the DAFO base, and and a trait database. And I said, this is quite crucial that you have access to traits, and this is also under development. But when we combine soil biodiversity information from those databases, we can again arrive at the ecosystem services. Finally, let's show you an example of how it can be done, where we have taxonomic information about earthworms, and that would be provided by eDNA, the species list at least, but not the biomass, as I said. And then we can go through the next step of assigning functional traits to the species, and we can then assess the processes that certain species with certain traits are able to do in the ecosystem. And finally, when we talk about earthworm, you can see here you have the earthworm casts or excrements, which is actually the way that soil are formed. And we can put a value on that because people will actually buy soil to improve, for growth of plants. So this can be a way to actually arrive at ecosystem services, which should be valuated. I mean, that's the whole concept behind ecosystem service, that is that you can put a value on it. So this was my words. Thank you.
Well, thank you, Paul. Thank you so much. It was also lovely to see the different connections between taxonomic groups, the different ecosystem processes and functions, and finally the other part of the ecosystem accounting as well, connected to it with the information that we can extract from soils. Well, now I would open here some space for questions and comments. I've already asked that in the chat here. Well, I can launch the first question. Paul already mentioned some methodological challenges that need to be addressed with this type of technique. Of course, here we saw the many opportunities that they are associated with eDNA. But I would also love to hear from the speakers the main difficulties and especially the main limitations, and how you see those limitations being tackled over the next years to make eDNA even more useful. Should we now start? João, maybe you.
Yeah, thank you, Sofia, for putting me in the spot immediately. Yeah. So obviously, because we've been looking at that and we recognize there's still limitations. I think we've seen some of them. Paul mentioned some, Karen as well in her slides, Zach as well. So especially sampling is a limitation still. Being able to create a representative sample, a composite sample that represents an ecosystem is still a challenge and requires careful design, and we are really aware of that when we propose new sampling approaches. Also, I think the quantitation aspect of eDNA and this, what Paul mentioned, the reads to species and being able to do it across the board is still a challenge that everyone, and is like the holy grail of eDNA that everyone is looking at. I would highlight those as the main difficulties at the moment. I would start with those. I'll leave the floor for the others. I won't run them all over.
Yeah. No, I'll just jump in and agree. In Brazil, particularly where TerraBio is working, we've had significant gaps in our taxonomic data set database that have definitely caused some issues, and figuring out how to work around those and still come up with a good product and informing KPIs and things like that has been definitely a challenge, but it's an exciting one.
I'll post just, if you allow me, just another one that follows from Karen, which is actually the lack of genomes that we still face. So we can do all this, and in Europe we're really well, in the US as well, and most species and most philanthropic projects are finest academic work to work on putting this genomes in databases, so we have barcodes and all that. But as Karen mentioned now, the Amazon, for example, and other remote locations, Africa, we've recently seen only 3% of species in a way represented in database. So this is a challenge, and for us that rely in a way on public available data, this is probably another big challenge.
Well, thank you so much, João and Karen. We have here Sarah that raised her hand. Sarah, do you want to interview?
Yes. So this is basically a question to everyone that works with insects. So we know that there's a major gap in terms of databases that cover this large groups of insects, but there's even a larger gap when it comes to what they do in the ecosystem, what their role is exactly. So how you see this moving forward in the future? Do you see that these gaps will keep be filling in, because we are still lagging a lot here when it comes to knowing what exactly an insect is doing? So do you see a lot of academics filling up these gaps, a lot of movement in this area? How do you see? This is a question for everyone that it's working with arthropods in general.
So I can start off if that's all right, because I primarily work with insects and arthropods. And so I fully acknowledge your 2 points. I would say, yes, the databases, the barcode databases are woefully incomplete, especially in certain areas, and B, even more so what do they do, their life history. And so I do similar things to Paul, but with arthropods, where we're getting traits and linking them to these rich taxonomic databases we get from eDNA. And I would say that, you know, in a sense, to me, I mean, they're two independent problems, but when you think about it, the problem of the traits or what they do, that's not something that is a unique problem to DNA. That's a problem that we just have because, frankly, since the 70s and 80s, there's less funding available to figure out what specific insects do, because we've moved, rightfully so, to some degree, we've moved to these global large-scale studies. And so we're missing a lot of that. And I don't honestly know how that is going to be, the trait part is going to be fixed in the near future. The more promising one for me is getting barcodes in these libraries to flesh them out. Every time I go give a talk, people are like, well, but the barcode libraries are not complete. And I would agree, but then counterpoint with that a couple points. One, they're getting better each day. Two, in areas, as several of you mentioned, in areas like Europe and certain areas of North America, they're really good. Like, they're really good. Three, do we necessarily need to know their name to know that it's biodiversity? And that's one thing with the eDNA, we get records, we just may not know what it is. And then just to circle back to what I talked about in my talk, this is where citizen scientists can be hugely helpful. Imagine doing a mothing event in Brazil. If we got in the middle of DFW, one of the biggest urban areas in the United States, if we're getting hundreds and hundreds of species in a pretty, in a tiny patch, essentially, imagine what you could get in Brazil. And because the cost of the barcoding part of this is cheaper and the barcoding part is a lot easier technically than the meta barcoding, this is just a matter of getting citizen science together and forming partnerships. But I think that if there was like some kind of funding initiative or global initiative, we could make huge strides in that way more quickly than we think.
Thank you, Zacchaeus. Paul, you had your hand raised.
Yeah, I'm just I can just more or less repeat that within the soil fauna, for instance, databases for traits are existing and are being developed. So I think there's quite a lot of attention to that. It's kind of along, eDNA and things like that. It's kind of a buzzword and it's also driven from the paradigm of being able to link traits to ecosystem services. So the ecosystem services, the concept actually drives that we need to know something about what they are doing in the ecosystems, right? So I mean, it's an ongoing process, and we should not forget there's actually quite a lot of knowledge and information about what species did before we spoke. People started to speak about traits. We still had nice, you know, life form descriptions, so on, which are quite useful, I would say.
Yeah, that's an interesting point, Paul. Exactly how to, and also as launched by João and Zacchaeus. And there are many gaps to be filled in, but going for the traits and the functional groups can actually be a great advance for, especially for the ecosystem services and to understand the ecosystem functioning associated. Well, we have here some questions written. This one goes for Zacchaeus about the citizen size is often connected to bias given by oversampling of attractive species or places that are close to populated areas. Could you comment on those perceptions and observations very briefly, Zacchaeus?
Yeah, that's a challenge for me, but I'll be succinct. I would say, yeah, I agree, of course. And so like, you have to think about how you're employing the citizen scientists. And so if what we want is to enhance the coverage, like we can enhance the coverage in your local area with the BioBlitz, but we can also think about, like I saw in the chat, somebody said they did these mobbing events in Brazil and they're getting huge amounts of taxa. And so we could, you know, there's citizens all over the world. Like we've got partners in remote areas of Canada where we know nothing about aquatic insects, for example. And so strategic partnerships can go a long way for that. Like there's most people we talk to when we do these citizen science projects, they're very eager to learn, and they feel like they just don't feel like there's access or that they could do science. So we can easily break down those barriers and then be strategic about where we do that, and that can help. And of course, citizen scientists are not going to do all parts of the eDNA pipeline, but they can be very much involved on the collection, which is one of the more costly and time-consuming parts. And then they can also be brought back in on the back end, where we could show them a picture of the biodiversity in their backyard and then what it means. And so, yeah, I'm very hopeful about this, despite it being, yeah, it's a criticism, but it just speaks to how we need to network better. Thank you,
Zacchaeus. We have here a question from João. I'll try to address this. This is actually also a curiosity of mine. So we see from these eDNA samples, and we manage to evaluate if a species occurs or not, or estimate that. But what about quantitative data, for instance, abundance? Or how do we know if the species that was detected is still alive, or if it is still present in the studied habitat? Who wants to take this challenging question?
This is at least a 20-minute discussion.
Is it possible to get from eDNA, or do you see it as a possible way to do it in the next, over the next years, I would say?
Well, I might put aside the abundance question, because that gets into a lot of very technical detail in terms of, well, anyway, I'm going to put that aside. It's a very technical discussion that might be too long. In terms of whether or not a species is still alive and whether it's, you know, we find things in the soil. And, you know, I've also done work with fungi in addition to the current work I'm doing with arthropods. And we've tested some mammal stuff as well. And I'll counterpose two questions. The first one is, how much does it matter? Because for some taxonomic groups, what you're looking for is that the animal transited through. So yes, you don't necessarily know, but you don't know with some other things or some other techniques as well. So the question, like, if it's very important to find a specific thing in the place and observe it with your face, you know, eDNA is not necessarily the right approach. But really what we're interested in is what is there that is leaving, you know, traces of its scales and excrement and things like that, that means that it is using the environment. And so I guess that's kind of a bigger question. I will also mention in different environments, DNA breaks down at different rates. So it's dependent as well on your particular environment. Working in tropical or very humid environments, it breaks down pretty quickly. So
thank you, Karen.
sorry.
João and Zacchaeus, you also have your hands raised.
I think Zach was there first. I think Karen said it all, basically. I said the quantitative, I think there's that connection between the stochastic nature of the sampling itself. So there's many things that come into play, and we would stay here another hour on that. But also, I think another thing, and I think that's, and we are here between many academics as well, what is clear as well is for some of these answers, probably there's even another complication. eDNA is probably not the answer. There is eRNA as well emerging in the literature now, and that is going to get a life of its own. And that's probably going to answer some of the questions that were debated here. Many different angles to approach this. Yeah. Regarding quantitation, what we're doing and how, as a company, because we have to do it sometimes, is really we are only when we're specifically talking about targeted approaches using PCR. That's the only times we're even considering such an approach.
Thank you, João. Exactly. Katie was also here mentioning in the chat about eRNA as a promising approach. This is new to me. This is nice. Zacchaeus, just a final comment.
I'll just add two brief points to what Karen and João have said. And that is one, like, I think people are always going to be interested in, can we get abundance and biomass from the data? And I think Karen's got a good point, like, do we need it? And I think a lot of times we don't, but certain government monitoring plans require by law some of this information. So it's going to remain a holy grail. So here are a lot of people are working on this, and we don't have time to get into that, but I would point you in two exciting new directions where people are trying to think creatively about this. The first is, I think in the future, like when we get better and better sequencing technologies, we will be able to do what we call shotgun sequencing. Instead of sequencing just small bits of the genome or the genomes in our sample, we could sequence the whole thing. And then we get away from PCR bias, which perhaps will give us better correlations for biomass and abundance, especially if we have trait information on how large these organisms are. So there's that angle. And then the other one that myself and some collaborators have taken is that if we think of it from a modeling approach. So if you repeat your sampling, so if you have multiple samples taken in the same environment, replicated sampling, you can do something called occupancy modeling. And so you can actually get a sense of abundance just by repeated presence-absence sampling. And in doing that, we've been able to estimate abundance pretty well, actually, in these initial studies that we've tried. So there's a lot of interesting ways that I think this will move into the future. But again, we could have a whole seminar just on this one subtopic.
Yeah, thank you so much, Zacchaeus. Well, people, we would have many other things to continue this discussion, but we are over our time. Well, thank you so much for everyone that attended. In case of any additional comment or question to address to the different speakers that were here today, I'm posting here in the web chat the announcement of this webinar, you can click on the different speakers and you will have their specific information and contact details. My special thanks to the Secretariat again, and especially Katie and Zacchaeus, João, Karen, and Paul for your availability. What a nice way to conclude the week. I'm sorry for those that are starting the day now, but we are done on this side of the world. So thank you so much, everyone, and have a lovely weekend.
Thanks,
Have a
everyone.
good weekend. Bye-bye, everyone.
Good weekend.
Bye-bye, hi.
Thanks.
Bye.